It's Michael and good day everyone. Today we are going to be discussing about carbohydrates. Carbohydrates is a class of organic compounds that contain carbon, hydrogen and oxygen, in a 1-to-1 ratio, and it comes with the basic formula molecule CH2O. And then the chain length of carbohydrates varies from few sugars to hundreds of sugars. So I mean to say carbohydrates that has longer chain, it consists of hundreds of sugars. Should be the longer the chain length, the more the content of the sugars. Carbohydrates are divided into monosaccharides. Meaning to say that particular carbohydrates contains only one saccharide molecule. Then we have disaccharides, which contains two saccharide molecules. Di means two, having two. And polysaccharides, meaning to say that particular carbohydrate contains many saccharide molecules. Now monosaccharides, they are simple sugars. Or you can simply say, monosaccharides are the monomer of carbohydrates. So meaning to say, monosaccharides, they are the basic unit of carbohydrates. These monosaccharides can be widely found in your kitchen or in your house. The ones that the sugar that you add to your cooking or to your drinks, those are monosaccharides. So single sugar molecule that cannot be hydrolyzed to simpler compounds. Classified into trioses, meaning to say trioses are monosaccharides with 3 carbons. Now class trioses, they have another word for this. Triose sugar. So trioses or triose sugar, it gives the same meaning. Alright. Now monosaccharides with 4 carbons are known as tetroses. And then we have pentoses, hexoses, and so on, according to the number of carbon atoms and type of functional groups. Now monosaccharides have a large number of hydroxyl groups. You still remember hydroxyl groups, OH. And because of these OH groups, these hydroxyl groups, it makes monosaccharides soluble in water. Now class, why does having hydroxyl groups makes the monosaccharide soluble in water? The functional group OH, it tends to form hydrogen bond with the water molecule. So when it tend to form hydrogen bond with the water molecules, it makes them soluble in water. Then these monosaccharides, they have carbonyl group, that is terminal. The location is terminal in aldehyde sugars, and located in an internal position in ketones. So meaning to say, monosaccharides, that are aldehyde, that are known as aldehyde sugars, if they have the location or position of its carbonyl group, at the terminal of the sugar. If the position or location of the carbonyl group, internally in the monosaccharides, these monosaccharides are grouped into ketones or known as the ketones. So we have ketones and aldehyde sugars, which both can be grouped as monosaccharides, right? Now this carbonyl group, C double bond O, is a functional group that gives monosaccharides sugar reducing properties. So you can see these aldehydes here, and ketones here. This is the carbonyl functional group here. Now each classes, of these monosaccharides, the sugar in the monosaccharides, they are structural isomers, meaning say they have same molecular formula, but they have different atoms arrangement. So because of the different atoms arrangement, their structure, okay, a bit of different, okay, from one monosaccharide into with another monosaccharide, okay, although they have the same molecular formula, okay. Now example is glucose, okay, and glucose is a hexose sugar that came with the molecular formula of C6H12 or 6, okay, and apparently this glucose is the common form of monosaccharides, okay, and it has fructose and galactose as isomers. So now whenever you see, okay, glucose, fructose and galactose, these three monosaccharides, okay, they have same molecular formula, okay, but different atoms arrangement. Okay, now glucose is the major source of cellular fuel for all living organisms, okay, and then readily broken down and converted into ATP during cellular respiration, right? Okay, so this diagram here, okay, shows you a glucose molecule, okay, sorry, a glucose molecule in its ring form, okay. Okay, now, sorry, not really glucose. It can be glucose, fructose, and galactose. Okay, so apparently this is the ring form of a hexose sugar. Okay, so this is the um, aldehyde. Okay, no, sorry, this glucose here, all right, this sugar here is an aldehydes. How do I know? Because the position of this carbonyl group, which is in terminal, okay. So now this, since this is a ring structure, okay, this ring structure here, when under IUPAC, okay, in terms of naming, okay, naming um, the position of the carbon in the ring, okay, it is clockwise position, all right, and clockwise position from the functional group, such from the functional group. So since this is the carbonyl functional group, okay, and this is the adjacent carbon, so this will be carbon 1, carbon 2, carbon 3, and so on, right? So now ribose and deoxyribose are examples of pentoses, okay, and are found in the nucleic acids RNA and DNA. Pentoses and hexoses can exist either in straight chain or ring structure. Okay, class, now these are the examples of aldoses, okay. Aldoses means sugars that has aldose, okay, sorry, that has aldehydes, okay, and these are the ketoses, you say sugars that has ketones. So aldose and ketones, they are only different because of the position of the carbonyl functional group, okay, terminal or internal. Okay, so as you can see from here, the location of this carbonyl group is terminal. Okay, so that makes this particular sugar an aldose. Okay, and the position of this carbonyl for this particular sugar is internally. So this makes this sugar a ketose sugar. Okay, now since this sugar here, okay, or to be more specific, glyceraldehyde only have 3 carbons, okay, so this particular sugar is a triose sugars or trioses, okay, so we have triose sugars here, pentose sugar here, okay, and then we have hexose sugar here, okay. Now, again, just now I mentioned about structural isomers, right, between glucose, galactose, and fructose. Okay, so if you look at this table here, okay, these three sugars, they have the same molecular formula, which is C6H12 and O6. The only difference is their atoms arrangements, which gives them different structural uh, different structural, right? Different structure. Okay, and this structure here is in the form of a straight chain. Okay, do you notice the position of this OH here? OH functional group. Okay, for glucose is on the right plane, and for galactose is on the left plane. And then the position of the carbonyl functional group as well. Okay, if it's terminal, so it's either glucose or galactose. If it's internal, it's um, ketose. All right, now, okay, triose sugar. Okay, this is how they look like. Okay, then this is the tetroses. Okay, triose and tetrose, they can't exist in ring form because they don't have enough carbon to form a ring. Right, for pentose and hexose sugars, they can form, they have a ring form, right, because they have enough carbons. And why being a ring form? Okay, because this ring form, apparently it gives them more stability in terms of their distribution of the electrons. Okay, right, ribose and deoxyribose, you can see for deoxyribose there's a missing oxygen atom at the carbon number 2 here. Okay, so because deoxy, De means de removal or missing, oxy, oxygen. So missing of oxygen at the second carbon here. All right. For pentose, you can see there is an oxygen atom. Okay, at carbon number 2. Right. Okay, so again you can see how in the in the structural form, in the straight chain form, how these ribose and deoxyribose, they are different because of the either present or missing of the oxygen atom. Okay, at carbon number 2. All right. Okay, now this glucose, okay, they are quite special. Okay, why this glucose is quite special? Because when dissolved in water, this glucose apparently they will have rearrangement of its atoms. Okay, and then because of this rearrangement of these atoms, it forms two types of glucose, either alpha glucose or beta glucose. Okay, now how do you differentiate which one is alpha, which one is beta? Okay, now apparently an alpha glucose, okay, it has the OH functional group, okay, on carbon number 1 under the plane. Okay, this is the plane. Sorry. Okay, this is the plane. It's under the plane. For beta glucose, the position or location of the OH functional group, okay, for carbon number 1 is above the plane. Okay, so whenever you see um, sugar, okay, to be more specific, glucose that has the position of the OH functional group, okay, at carbon number 1 above the plane, so that would be a beta glucose. All right. Okay, now another thing is, okay, um, D-glucose and L-glucose. Apparently this glucose, okay, when they are the alpha and beta glucose, they are optical isomers or stereo isomers. Okay, so for D-glucose, okay, the OH functional group at carbon number 1, 2, 3, 4, 5 is on the right side, and if it's on the left side, it's known as L-glucose. Okay, and take note class, okay, this is the reducing end, the carboxyl functional group, okay, which give this glucose the properties to reduce the Benedict's solution. Okay, and apparently this is the non-reducing end, which does not reduce the which has no reducing properties. Okay, so I'm not going to further explain about this, okay, since I already explained to you. Okay, now monosaccharides have the flowing properties, okay, because of the presence of the hydroxyl groups. They tend to be sweet, okay, polar, are soluble in water, and then form white crystal readily. That's why your table sugar or the sugar in your kitchen, they appear to be in white crystal. Okay. Okay, next, disaccharides. Okay, disaccharides are sugar formed from two units of monosaccharides by condensation, or known as, okay, other word for condensation is dehydration. Okay, because condensation and or dehydration, it involves removal of water molecule. Okay, now the bond between the monosaccharides, okay, is known as glycosidic bond or glycosidic linkage. Okay, and this bond that is between the monosaccharides is formed between the carbon 1 of one molecule and carbon number 4 with the other molecule. Can you imagine you have two monosaccharides, okay, and there is a bond between them, which is known as glycosidic bond, and this glycosidic bond is formed, okay, from the first carbon of the first monosaccharide and on the fourth carbon of the other monosaccharide. Okay, physical properties of disaccharides, okay, sweet, soluble in water, and form white crystal readily. Okay, so apparently more or less with the monosaccharides. Okay, why does disaccharides have sweet, um, soluble in water, and form white crystal readily as physical properties? Why? Can you give me one possible or one reason why? Because of its many hydroxyl functional groups, okay, which are able to form hydrogen bond with the water molecule. Okay, chemical properties for disaccharides, they are all reducing sugar, okay, except for sucrose, because sucrose apparently they don't have reducing end. Okay, now example of disaccharides, we have maltose, sucrose, and lactose. Okay, sucrose is the form in which sugar is transported in plants by translocation. Okay, we can acquire sucrose from plants such as sugarcane and sugar beets. Alright. Okay, so this is how sucrose is formed. Okay, it's a disaccharides of glucose and fructose, and this is the glycosidic bond or glycosidic linkage. Okay. Okay, so this is the first carbon and this is the fourth carbon. Okay, now lactose, okay, is a disaccharide that can be found in milk or dairy products. Lactose is glucose combined with galactose. So can you get that picture? Okay, glucose and galactose give you lactose. So individuals that are lactose intolerant cannot break this disaccharide down, okay, which leads to gastrointestinal discomfort because you lack of this particular and one particular enzyme that cannot break the lactose into glucose and galactose. Okay, now this is the dehydration and degradation of maltose. Okay, maltose is formed when you have two glucose. Okay, you can see here how this OH and H, OH from the first monosaccharide and hydrogen from the second monosaccharide. Okay, they are being removed as water, and they form this alpha 1,4 glycosidic bond. Why alpha 1,4? Okay, now the name of this bond is, this is glycosidic. Okay, it tells you this is a glycosidic bond. Okay, now alpha, why alpha? Because of the position of the OH on the first carbon, which is under the plane. You can see from here it's under the plane. Same goes here as well, because we have two glucose here. So the location of the OH here on the first carbon must be under the plane. So this is an alpha glucose. So alpha 1,4 means the bond, the glycosidic bond or glycosidic linkage is formed between the first carbon here, right, with the fourth carbon of the other glucose molecule. 1, 2, 3, 4. Okay, first, second, third, fourth. So first carbon with the fourth carbon. So alpha 1,4 glycosidic bond, right? And in the IUPAC, okay, in terms of naming, naming of compound, molecule, or linkage, okay, between alpha and any digits, there must be a dash. Between digits, with digits, there must be a comma. Between digits with letters, okay, or alphabets, there must be a dash. So alpha 1,4 glycosidic bond. All right. Okay. And this diagram here tells you, okay, the degradation or hydrolysis of sucrose. Okay, you can see, okay, the glycosidic linkage of this sucrose is being break, and then you have glucose as well as fructose. Okay, now can you tell me what is the name of this linkage? Okay, is it alpha or beta for sucrose? Is it alpha 1, 2 glycosidic bond, or alpha 1, 2 glycosidic bond? Okay, or beta 1, 2 glycosidic bond, or beta, okay, 1, 4 glycosidic bond. I'll leave that question to you. Okay, you can come over and ask me if you still can't figure it out. Okay, again, let's recap. Monosaccharides are simple sugars. Disaccharides, there are two monosaccharides units, sugars with two monosaccharide units. Oligosaccharides is sugar or carbohydrate that has 3 until 10 monosaccharide units. Okay, for polysaccharides, they are carbohydrates that has more than 10 units of polysaccharides. Okay, and polysaccharides can be further categorized into homopolysaccharides and heteropolysaccharides. Okay, now polysaccharides, they are the most abundant carbohydrates. Okay, they are complex carbohydrates or macromolecule, being said macromolecule means to say polysaccharides molecule is huge. Okay, macro, which is the opposite of micro. Okay, made of repeating units of monosaccharides, maybe a single long chain or a branch chain. So I mean to say polysaccharides, when you see them structurally, okay, if they are long chain, okay, they are unbranched. Okay, if they are shorter chain, they exist as shorter chain, they are going to be branch. or highly branched. Okay, two types of polysaccharides. Homopolysaccharides, meaning to say all the constituent monosaccharides are of the same type. Okay, example of homopolysaccharides are glycogen, okay, starch, cellulose, chitin. Okay, these are homopolysaccharides. Then heteropolysaccharides, okay, they are polysaccharides that have more than one type of monosaccharides. Example, peptidoglycans, okay, glycosaminoglycans. Okay. Oh, physical properties of polysaccharides, they are not sweet. Okay, tell me, starch. Do starch taste sweet? No. Okay, unless the starch is digested, then it can give out its monosaccharides, which is the simple sugar, right? Monosaccharides are simple sugar. So when starch are digested, okay, into or break down, when starch are break down into simpler components, okay, the simplest component will be monosaccharide, which is sweet. Then it will taste sweet. But as a polysaccharide, they don't taste sweet. Insoluble in water, okay, cannot be crystallized, okay, but they will form amorphous mass. Okay, meaning to say this amorphous mass means to say is sort of like gel, like a gel. Okay, and yes, and it doesn't have any definite form. Okay, definite form means to say bentuk tetap, compact, okay, and then osmotically not active in cells. It doesn't allow, it doesn't not it doesn't allow, it doesn't causes osmosis. Okay, and then extracted and purified to form white powder. Okay, have you heard of cornstarch before? Yes. Okay, that is an example of polysaccharide. Cornstarch, the starch that is um, in powder form, white powder form. Okay, so apparently those are the cornstarch in the kitchen. Okay, you can ask from your mom if you have no idea what is cornstarch I'm talking about. Ask from your mom. Okay, they exist as a white powder because they are already extracted and purified. Okay, and then you may try put some on your tongue, the cornstarch. Okay, and then tell me what was your initial taste of the cornstarch. Okay, it doesn't taste sweet, trust me. Okay, chemical properties, they have no sugar reducing properties, okay, power, can be hydrolyzed. Okay, starch and glycogen, okay, are hydrolyzed to maltose by amylase. Okay, then cellulose can be hydrolyzed to glucose by cellulase. Okay, now um, example of homopolysaccharides like glucan, fructan, galactane, arabian, xylene. I'm sure you've seen this, okay, behind the label of your food. Okay, so glucan means say is a polysaccharide of glucose, fructan, a polysaccharide of fructose, right? So on. Cellulose, okay, on the other hand, is an unbranched homopolysaccharide made up of glucose monomers that is connected via beta glycosidic bond. So now, can you imagine cellulose? It is a homopolysaccharide that is in a long chain. Okay. All right. This is to give you an idea, okay, of homopolysaccharide that is unbranched. You can see there's a long straight chain. An example of this is the cellulose. And then if they are branching out, they appear something like this. Okay. Now for heteropolysaccharide, if they are unbranched, okay, they are like this. They consist of many different types of monomers, okay, of monosaccharides. Different types of monosaccharides. And if they're branching, something like this. Okay. All right. Now let's go to starch. Okay, class. Now starch. Okay. It's a typical form of carbohydrate. Okay. and starch are used as energy storage in plants. Okay, and starch is a polymer consisting of alpha glucose subunits. Okay, so since starch consists of alpha glucose subunits, so meaning to say the bond is between these glucose subunits are known as alpha glycosidic bond. Okay, now these monomers are joined by alpha 1,4 linkages, which means that carbon 1 of one glucose is linked to carbon 4 of the next glucose in the chain. Is it? Let's take a look here. Okay, this is the first carbon, alright, and this is the fourth carbon. And how do I know this is an alpha glucose? How do you know this is an alpha glucose? Look at the position of the OH on the carbon number 2. Okay, both are under the plane. Okay, so it's alpha 1,4 glycosidic bond, right? So these are all alpha 1,4 glycosidic bond. Okay, now starch occurs in two forms, amylose and amylopectin. Amylose is a simpler form of starch. Okay, why? Because they are unbranched. So apparently this diagram here, there is no amylose here. Okay, these are example of amylopectin. Okay, now amylopectin is a more common form of starch, okay, and usually consists of about 1,000 glucose units in a branch chain. You can see this first chain here, okay, it branches, okay, over here with this another chain here, okay, and these two, okay, chain, they are connected by this bond here, okay, between the first carbon of this chain, okay, this particular glucose with 6 carbon here, okay, and this link here, this bond here is known as alpha 1,6 glycosidic bond. Okay, alpha 1,6 glycosidic bond. Okay, now when they branching off, right, okay, they branching off at this point here, right, which is known as branch point, the branch point. Okay, and then since they are highly branched, so what happens, okay, to give them stability, okay, the chain beyond the branching point, they will coil up, okay, they will coil or appear in a helix form. Okay, you can see here, this is helix or coiling. Okay, now this gives them stability. Why? Because when they are coiling or helix, okay, or I said they structurally in a helix form, okay, what happens, the hydroxyl groups, okay, between one unit with one unit, okay, will form hydrogen bond. So I give you one example here. Okay, let me change the color pointer to red. Okay, so the hydrogen bond, okay, would be between this oxygen, okay, for example this hydrogen here, okay, they form hydrogen bond. Okay, so the same goes for this hydrogen here, you will form a hydrogen bond with another oxygen from a OH functional group of another glucose unit here. Okay, a hydrogen bond gives stability to the starch. Okay, let me change it back to yellow. Now, amylose is an unbranched polymer, okay, right, with one alpha 1,4 glycosidic bond. Okay. Now class, amylose, okay, although it is a straight chain, okay, this is something which I did not mention beforehand earlier, okay, they are coiled, okay. Why are they coiled? Okay, to form the alpha helix or the helix structure. Why? Again, to give them stability because of the hydrogen bond, okay, between hydrogen and um, hydrogen of one glucose unit with another hydroxyl functional group of another unit. Okay, they are in one straight chain like this, but it's coiled. Okay, it's coiled. Okay, now amylopectin is a branch polymer with alpha 1,4 and alpha 1,6 glycosidic bond. Now class, there is a dash here. Okay, I think there's a typing error here. There's a dash here. Okay, and this should be a dash here, another dash here, and then glycosidic bond here, right? And there should be a dash here as well. Okay. Okay, can you see the difference between an amylose with an amylopectin? Okay. Okay, so apparently this is starch granule that can be found, okay, inside a potato, apparently. Okay, so physically this is how they appear, and in terms of molecular level, this is how they are. Okay. Now cellulose. Okay. Remember earlier I said polysaccharides. Okay. Homopolysaccharides. Examples are starch, cellulose, and glycogen. So we are going through these three. Okay. Examples. So cellulose, an insoluble, okay, polysaccharide composed of many joined glucose molecules. Cellulose contains beta glucose monomers, right, joined by beta 1,4 linkages. Okay, now these beta 1,4 linkages cannot be split up or broken down by enzymes that hydrolyze the alpha linkages in starch. Enzymes, so we need to say enzymes that hydrolyze the alpha, okay, and the enzymes that can hydrolyze the alpha linkage cannot hydrolyze the beta linkage. Okay. Now, humans, like other animals, we are lack of enzymes that digest cellulose. Okay, the cellulose found in whole grains and vegetables remains fibrous. Okay, and provides bulk that keeps our digestive tract to functioning properly. Okay, so they are good side as well. Why we are lack of the enzyme to digest the beta 1,4 linkages. Okay, otherwise all the fibrous food that you take will be digested, so it doesn't give you um, bulk. Okay, and this will eventually leads to constipation. Okay, remember, taking fibrous food will prevent you from getting constipation. Some microorganisms digest cellulose, okay, to, to glucose. Example, cellulose digesting bacteria which live in the digestive system of cows and sheep. This will enable, okay, these grass-eating animals to obtain nourishment from from cellulose, right? The digestive systems of termites contain microorganisms that digest cellulose. Cellulose molecules are well suited for structural role. So meaning say cellulose, okay, their molecules are when it comes to functions, okay, they are more on structural role. They give structural support. Now, this is because, okay, the beta glucose subunits are joined, okay, and allows extensive hydrogen bonding, okay, or cross-link among the different cellulose molecules. Okay, it's very similar to the ones in the polysaccharides that I've explained to you earlier. Okay. Okay, now this is to show you that the cellulose molecule is unbranched polysaccharides. Okay. Okay, you can see the interaction. Okay, not interaction, sorry, the hydrogen bonding. Okay, this is what they call crosslink. Okay, between the oxygen and hydrogen of two different subunits of monomers in the cellulose. Okay, on different chain, how they form hydrogen bond between each other. Okay, do they branching? They don't. Okay, it's a long straight chain. Okay, so this diagram here show you cellulose fibers, okay, from a cell wall. Okay, now glycogen, okay, usually is being referred to animal starch. Okay, is the form in which glucose subunits they are joined by alpha 1,4 linkages. Okay, and stored as an energy source in animal tissues and readily broken down when energy is needed. Okay, glycogen is similar in structure to plant starch but more extensively branched or highly branched polymer of glucose and more water soluble. Okay, you can see here they are highly branching off. Okay, highly branched. Now in vertebrates, glycogen is stored, okay, in liver and muscle cells. Okay. Alright, this is to show you comparison in terms of structure between starch, glycogen, and cellulose. Okay. Alright, so okay guys, so that's it for carbohydrates. Okay. Okay, thank you. Bye.